What's up, Info Penguins? So we are live and we are ready to rock this exam. So in case you're just tuning in, of course, hi, I'm Mrs. Jones. I'm from AP Bio Penguins. And so I'm going to do a quick Q&A session over just AP Bio. I asked the past 2 days for students to submit questions to me. And when they submitted those questions, I then compiled them all. I thought that 5 hours was going to be enough time to compile all your questions, but that was not enough time. So I have gotten through 4 units. So my Q&A session today has a little bit of just kind of like exam questions people had, and then some like science practice questions people had. But then I'm working through unit 1 through 4. And if you hear any random noise in my background, it's my children. Just ignore them. And so we're going to go through units 1 through 4. And I'm going to do whatever I can. I'm going to answer whatever questions I see in the chat as we go. But remember, I am one person. I'm human, and I'm doing everything I can to, appropriately prepare you for the exam that's happening in 8 days. So as a quick reminder, just, who am I? I'm Mrs. Jones, and I am AP Bio Penguins. And I mean, like, I am AP Bio Penguins. There is no one else that works or does anything on AP Bio Penguins. It is just me. So whenever you send a message on Instagram, whenever you send a message on YouTube, whenever you send a message in any way, shape, or form, it is just me here trying to help you in the best way that I can. So there's a ton of resources. If you go to apbiopenguins.weebly.com, there's, like a ton of resources. There's a 396-page review guide. There's an Instagram review that I did every single day. You've got the PowerPoints that go with that. I've got 120 quizzes, games. I've got, a CRAM session. I've got anything that you could possibly want is posted on that website for you. So just go there. And then, of course, my Instagram is AP Bio Penguins in case you want to follow it or, get last minute review stuff. So as I said, I only got through 4 units. It's 100 and something slides, but I got through only 4 units in terms of the Q&A. I'm going to do everything I possibly can to get you everything I can, but I am human and I'm answering the questions that were submitted. There is no rhyme or reason. I did them in the exact order they were submitted. So you're going to see that we are going to jump around a little bit just because of the fact that I didn't even have a chance to put them into a logical order. I finished honestly 10 minutes ago and said, okay, better change my clothes and get online. So if you're needing more of a lecture instead of just this Q&A session, I did a session last year. It was a week before the exam. And so it's a 2-hour session. I'm so sorry. But it is a cram session. It is a speed run. Those of you who like to speed up your person, you can if you try to speed me up, you're welcome to, but I mean this 2-hour thing that I did is a speed run. It is like fast as fast can be, but I went through all the units. I go through some practice questions. So I'm really last year was the cram. This year is the Q&A. So I'm going to try to take my time to explain the answers. But again, it is 100 something slides. So I'm going to try my best to get through this in as close to an hour, but it is what it is. And so I'm going to finish my PowerPoint. So however long that takes, I will finish my PowerPoint because I promise you I will finish. So just quick overview. On May 5th, Cinco de Bio, you are taking the AP Bio exam. There are 2 sections to it. And as you don't know, you are going to be hybrid. So that means that you will have your computer and you will open up the computer for section 1 and you will answer the questions on the website, or not the website, on Blue Book for section 1. It is 60 multiple choice questions and you have 90 minutes. So some students are always like, okay, what is the timing for this? Okay. So since you have, 60 multiple choice, 90 minutes, that's about a minute and a half a question. Some questions aren't going to take you that long to go through. You only need, maybe 30 seconds to answer the question or whatever. Okay. So on average, it's a 1.5 minute. Okay. So every 10 questions you get through, just check your time. Make sure only 15 minutes have elapsed. Okay. If more than 15 minutes have elapsed, you're going too slow and you need to speed it up, pick up the pace. Okay. If you have less than 15 minutes, you're doing a good pace. Keep on the pace you're going. And that will give you time at the end to go back and relook at any questions you might be not so sure about. Okay. You do have Blue Book, so you have the way that you can mark in there, like to come back for review, to check any of your answers. As you're working through the questions, my advice would be to go ahead and pick whatever answer you think is the correct answer. And any of them that you're unsure that you do want to go back to, just mark those for review, write them down on your scratch paper if you need to. Just because of the fact that with being on Blue Book. You're not going to be able to, oh, there's 1 minute left of the exam. Let me just quickly bubble these things down. You're going to have a different question on every page. It's going to take you a second to click or, move over to the different pages of Blue Book. So just go ahead and mark your answer. Even if you're not 100% sure of it and you want to go back to it, just mark that for review, and then you'll be able to go back if you have time. Then you're going to get a 10-minute break. If you've taken the SAT, your screen goes black, and it will have like a little countdown timer for your 10 minutes. It'll tell you to get up and go out and have your break. You'll get your water, you'll get your snack, whatever it is you're going to have during your break. And then after 10 minutes, you'll come back and you'll get section 2. Your books, as far as what I understand from the AP Coordinator's Manual, that you're going to already have the FRQ booklet on your desk. Yes, there's a timer on Blue Book. It will be counting down from what I can understand. So you will have a timer to be able to see at all times how long you have left on the exam. So section 2, you have 2 longs and 4 shorts. You also have 90 minutes for this. So the approximate timing you need, about 20 to 25 minutes for your long FRQs, about 8 to 10 minutes for your short FRQs, okay? And later on, somebody does ask a question on how I would pace that out, and I do go over like my pacing for that. So first question someone asked is, what units are typically the most covered on the exam? Okay, so this is from the CED. So in case you don't know what the CED is the curriculum. It is the standards that your teachers are using in order to determine what to teach you on the exam. So when you're trying to figure out what do I need to know for this AP Bio exam, just go to the CED and you will see everything that you are required to know for the exam. Now, the exam is going to be more applicational. It's going to give you a little bit more background information that you're going to need to have, but the actual content that you're going to need is right there for you. Okay, so based on this, you can see the weighting that we have for the exam. It looks like unit 3, 6, and 7 are going to be the most highly tested sections, but when you look at the grand numbers of it, it's only going to be a couple questions that are on these instead of the other ones. So it does seem pretty much balanced between all of them. Someone says how many scratch paper you get. You will get 5 papers of scratch. I believe they're giving them to you one at a time. They will not give them to you all at once, but you will have those scratch. Ooh, I forgot to mention something. So your questions for the FRQ will be on your Blue Book, but you are handwriting your responses. What I meant by hybrid is that you will be handwriting the responses in a book, and that book will be already on your paper. I don't believe you're allowed to have earplugs. As far as I understand, you shouldn't have anything in your ears for the testing. So somebody said, hey, Miss Jones, can you help me with the timing for this? Okay. Well, as I said, there's 90 minutes, 2 long, 4 short. So your timing, you click the button, you open up your exam, give yourself about 8 minutes and look through the questions. Figure out what questions is it that you know really well that you're like, hey, I got this. No, you can't have a ruler. What questions do you like, hey, I'm really good at cell respiration. Let me go ahead and answer these questions or whatever. Okay. And in your mind, renumber them, or on your scratch paper, whatever, renumber, determine what order you're going to answer them. Give yourself 1 long, 2 short, 1 long, 2 short. Okay. This ensures that you have enough time to get through all the questions, or as much of them as possible. Okay. And then you're answering what you know first. Okay. It's a points game. Your goal is to get as many points as possible in those 90 minutes. Okay. So if you really know something, go ahead and answer those questions first, and the things you're not so sure about, answer them last. So if you do happen to run out of time, it was the things you didn't know that you didn't get to, attempt for. So that's my option. Okay. So 8 minutes reading, 20 minutes you do the long. Then give your 8 minutes for the short FRQs, then 20 minutes for the next long, and then 8 minutes for the next short FRQs. And then that gives you 10 minutes at the end to be able to do any last minute additions to the questions. If you ran out of time and wanted to finish something, you can go through that. Yes, so the long FRQs are worth 8 to 10 points each. And so together the long ones are going to be 18 points. And then your short ones are going to all be worth 4 points. And so when you look at the questions, the long ones are going to be like A, sub I, and they've broken them down so you can see all the points. The number of questions you have, for the most part, tends to be the number of points. Other, like graphs, is going to be like 3 points. But other than that, like most of the things, if there is a specific question, there's a bolded term, traditionally those are the number of points you have. Okay. I don't know the percent you need. Just do the best you can on the exam. Okay. So you've seen that I've talked about, doing the FRQ in writing. So when you open up your book, it's very first thing talks the directions, and it says you may use pencil or pen. So I'm going to tell you my recommendation. I think you should be using pen on the exam. Okay. Every single portion of the exam should be written in pen. What if I make a mistake? That's okay. Just scratch it out, make a big X, and then keep going. Okay. It doesn't matter. Okay. You are welcome to make mistakes. If you need more paper, they will give you more paper. You do not have to worry about that. Okay. So use pen to write it. The only part that I would like you to use pencil for is your graph. You and I both know you're going to start graphing something, then realize you don't have enough space to put all the data on there. Or you're going to realize that you messed up one of the plotting of your points. It happens. We all do it. But with an eraser, you can erase that with your pencil, okay? But if you're using a pen, you won't be able to do that. So I recommend for using pen on everything except for pencil just on your graphing, okay? The pencil will scan. It doesn't scan as like, it scans well, yes, but it's not as like dark and as clear as the pen is. And so that's why I recommend that you do the pen for everything else, okay? So sometimes, this is my PSA, okay? Many years, this has broken my heart that I open up the scoring and I'm grading the exams, and then I see a person who's scratched out their entire paragraph long response. It could be a whole page. And then they've rewritten it somewhere else on the book. Okay. So please do not do this. Okay. You make a mistake. You wrote the question in the wrong spot. You've got this whole paragraph in the wrong spot. What do? Go to the question where it's supposed to be. So here we see question 3. We're supposed to be writing the response to question. Like, oh, I already wrote question 3. I wrote on question 2. Just write it out. See question 2. This tells the grader when they get the question, because it will pop up straight to the question they're supposed to be grading. It will go to this question, and then they're like, oh, wait, let me go to question 2. And then they can open full book and see your question and be able to answer, like, be able to grade your response. Okay. So do not freak out if you write in the wrong spot. Okay. It just makes it easier for us to be able to find your response. Okay. So someone says, why can't I use erasable pen? Erasable pen is going to erase in the heat. Your exams are on hot trucks moving all over, of course, the nation to get to where they need to go to be scanned. So I just recommend not using erasable pen because it could potentially cause it to erase. Okay. So let's get into unit 1. So unit 1, what's the difference between unsaturated and saturated fats? Okay. Now again, this is in no particular order. I'm answering the questions in the order that I got them on my Excel file. Okay. So unsaturated fats mean that they're going to have no double bonds. Okay. Every single carbon is fully saturated. It has hydrogens in every single spot. Okay. Versus your saturated is going to have a double bond. Okay. That double bond means that it's Actually, I said that backwards. Oh my God, that's a typo. This is what happens when I'm like quickly going through things. So my saturated is going to be the one that has no double bonds. My unsaturated is the one that has double bonds. I'm so sorry, y'all. So here you can see our saturated, as I said, has all these single bonds in it. And then the unsaturated, okay, is not completely saturated, is not fully, covered in these hydrogens. And here is that double bond. It could have more double bonds. It just has to have a minimum of 1 double bond on it, okay? And so what you're going to see is that these traditionally are going to have a kink. They're not going to be able to get as close together. And so we'll see when we look at like the plasma membrane and the phospholipids and how they have these fatty acids, they're going to cause it to get the kink, which causes it not to be able to get super close together, which helps with the fluidity of the membrane. Okay. So can I describe the different bonds? Yeah, so there's 3 different bonds you need to make sure you know. Yeah, the double bond is that kink that you see if it's a cis double bond. So describing the different bonds. Okay, so there's 3 main bonds you need to make sure you do know about. Covalent bonds, ionic bonds, and hydrogen bonds. Okay, the most of the bonds you're going to see will be covalent bonds and hydrogen bonds. That's really where the bulk of biology is. Okay, so covalent is going to be sharing valence electrons. So if you've ever taken chemistry, you know that there are these electrons that are on the outer shells, and so those electrons that are on the outer shell, if they're sharing them, that just means it's covalent. Their outer shells are going to overlap with each other, and by overlapping, that allows them to share it, so they each kind of finish their octet, if you may. Okay, so there's 2 types. There's a polar and there's non-polar. Polar means that you have unequal attraction. So there's going to be an electronegative atom and a non-electronegative atom, or less, actually, whatever. Okay. Anyways, so like oxygen is going to pull the electron a little bit closer to it, making it partially negative, while like a hydrogen doesn't pull it as close, so it's going to be partially positive. Okay. Versus nonpolar, you're going to have equal kind of pulling on the electron. Okay. And so you're going to feel like carbon-carbon bonds or carbon-hydrogen bonds. Those are going to be where you have those nonpolar. Okay. And that's going to be important when you think about like the solubility of things later on, or if they ask like, oh, is this polar, is this nonpolar? That's really what you have to go back to is it going to be a polar bond in there? Is there going to be a nonpolar bond? That's really where it comes down to. Polar tends to be our hydrophilic molecules, things that are water-liking. Nonpolar tends to be our hydrophobic and kind of water-fearing. Okay. Ionic bonds means that we have a transfer of the electron. So one atom has taken the electron from the other. So like sodium and chlorine, and those make ions, which will be important when we look at dissociation a little bit. Okay. And then hydrogen bonds. Hydrogen are going to be it's a type of it's due to the Hydrogen bonds are due to the polar bond. So the hydrogen is in a polar bond, so as I said, it's partially positive, and that's going to cause it to be attracted to an electronegative atom that's already bonded somewhere else. It is an attractive bond. There's no physical bond between the oxygen and the hydrogen. Yeah, the oxygen and the hydrogen, okay? They're just attracted to each other due to their partial differences of charges, okay? And so you usually see it with oxygen, sulfur, nitrogen. Those tend to be your kind of electronegative atoms that cause this. And when you think about your different macromolecules, you think about that there are different hydrogen bonds in those. And I realize I'm not even looking at the chat to see y'all's questions. So I'm trying. Whenever I see a question pop up, I am answering it, but I'm trying to make sure I get through these. So yes, you can bring your own pens and pencils to the exam. So how do I identify the R group is polar or nonpolar? So as a real quick reminder, R groups is dealing with the amino acids. And so you have to think about the bonding that you have of that R group. Okay, so we have the amine group, that's going to be the same for all amino acids. The carboxyl group, same for all amino acids. This hydrogen, same for all amino acids. The thing, the difference is that R group. So that R group is going to determine whether we have a polar or a nonpolar bond. Okay, so looking here, oh, somebody says, do I have to memorize them? No, you don't need to memorize them, okay? So, but you do need to make sure that you're looking at them to figure out it. Hydrogen can also, I think, go with fluorine, but you're not going to see that in biology. Okay, so with nonpolar, so here you can kind of look at this side chain, this little colored group, okay? Do you see how there's like mostly all carbon hydrogens? Like it's like carbon hydrogen. There's a sulfur here, but I mean like that sulfur has a carbon on both sides, so it's going to be equally pulling on both sides. So this also ends up being nonpolar. Yeah, there's this one little nitrogen here, but for the most part, everything is like carbon-carbon bonds. So this is showing you your nonpolar. Okay, and in terms of folding for our protein, nonpolar is going to fold inward. They're going to kind of hide from the kind of the water on the outside. Okay. Ooh, hey y'all, if you're taking AP Chemistry or you're going to be taking AP Chemistry, make sure you follow Chem with Corinne. She just chatted, she just wrote in the chat. She is phenomenal. She will help you all next year if you're taking it, or this year if you're taking it this year. Anyway, so polar. So if we look at the side chain for our polar, okay, look, I've got oxygen, 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 a double bond oxygen, nitrogen, nitrogen, oxygen. There's all these electronegative atoms in there, and that makes it polar. That makes it kind of pull that electron, okay? And then if we're looking at if it's charged, we're seeing, okay, is there a positive charge? Look, positive, positive, positive. Is it negative charge? Negative, negative. Like, y'all, seriously? And so the polar and then also the charged ones are going to fold outward toward the water because of the fact that it is hydrophilic, okay? They are water liking. They can create those hydrogen bonds with the water, which is what allows them to be, hydrophilic, okay? So next question, what type of bonds are we going to find in our macromolecules? So there was a question a couple years ago about sugar, and it asked about the bonds that were in the sugar. And a lot of students wanted to write glycosidic linkages. Well, the problem is the question was about in the structure. So if you're talking about in the structure, we're looking at covalent bonds. For the most part, everything inside of that structure is going to be a covalent bond, the bond that's attaching all these molecules together, okay? But if you're asking about the bonds between our monomers, that would be like the carbohydrate's going to have the glycosidic linkage. The protein will be a peptide bond. Nucleic acids will be a phosphodiester linkage. And then a phospholipid, I did put that on there, although that's a major bad typo because the fact that I was trying to get this done in the car, which clearly I couldn't see my screen. It's an ester linkage, okay? And that's just going to be between the hydroxyl of your glycerol with the fatty acid. And so these aren't, there's no monomer in a phospholipid, but that's why there's a star there, is just to tell you, hey, that's the type of bond, but you really don't need to know that too much, okay? But so in case you're trying to figure out the bonds, okay? Someone says, explain evaporative cooling. Evaporative cooling is how your body can cool down. So if you're really, really hot, your body's going to sweat. You're going to release all these, all this water out of your body, and your skin is hot. And so the water is going to be heated up by your skin. And so as you break those bonds between those water molecules, as you break the bonds that you have, those hydrogen bonds, as you break them, then the liquid water turns into a gas, and it will then, of course, leave your skin. And so you've taken the heat with it because that water absorbed that heat in order for it to go and, leave your skin, of course. And so that's what we see with evaporative cooling, and that's how we see with the vaporization of water and how that connects to it, okay? Somebody says, well, do we need to know about the bond that's between a random molecule? Probably not. That's more of a chemistry topic that you're going to have these random molecules have to figure out like how they're connected to each other, okay? Now, they may ask you to, here's this R group, tell me what is the polarity of this, and that might be possible. Or they may give you, a macromolecule and then ask you some polarity of it, okay? But for the most part, you're not going to be needing to do it. And most of the bonding that you're going to see in biology is covalent bonding, okay? I'm sorry, your classes are behind. Y'all, as I said, wait, did I skip that slide? Where did the cram session go? Did I not talk? Y'all, if I didn't say it, there's a cram session, it's a 2-hour session, like tons of resources. Anyways, what are the functional groups and what do we need to know about them? Okay, so in terms of your functional groups, okay, you need to, of course, be able to recognize the 7 main ones, okay? So carbonyl is a C double bonded to an oxygen, so CO. Your hydroxyl is an oxygen and a hydrogen. Your carboxyl is just a carbonyl and a hydroxyl bonded together, which then makes a C double bond O, OH. You have the carbonyl with the hydroxyl making the carboxyl. Your sulfhydryl is a sulfur and a hydrogen, so just like hydroxyl, but it has a sulfur instead of an oxygen. In case you've ever looked at the periodic table, you know the oxygen is directly below the sulfur, and so yeah, oxygen's above the sulfur, and so like they have the same kind of bonding masses. Okay. The phosphate is a PO32 minus. Clearly, I ran out of time and didn't make those subscripts. And then we've got the amine, which is NH3, and then the methyl group. Okay, the first 6 I mentioned are all polar. Okay, and the last one is nonpolar. Okay, so you see the phosphate with ATP, you see the phosphate with your, nucleic acids. You see the amine group when you're looking at your proteins. You see the carboxyl in your proteins. You see the sulfhydryl as an R group within your proteins. So these are just going to be interspersed within all of the like different macromolecules. Okay, so they may just ask you a quick question about them. So somebody's, I guess, asked about proteins. Yeah, there's an N terminus, which is where you're going to start your protein, and then it ends at a C terminus. So it goes N terminus to C terminus. And then in terms of like DNA, it's like 5', which is where your phosphate group is, to 3', which is where your hydroxyl of your sugar group is. Okay. So someone says, oh, that must have just gotten out of order. So evaporative cooling, that was a picture that was going with it. I was wondering where my picture went, but clearly it's right here. So when you're looking at like the skin, right? So the skin is heated up. We have, of course, that water on it. It causes it to evaporate. And then, of course, the cool air kind of replaces it, which is what kind of helps to cool you down. So there's the image to go with the slide I had like 4 slides ago. Clearly, I told you I was like last minute throwing this together in 5 hours for you. So explain water potential, solute potential, and pressure potential. I don't think this is a unit 1 topic. I think this is more of a unit like 2 topic, but like we talk about water in 1, so I just let it lie. So water potential, this has to do with pressure potential, pressure solute potential. Pressure potential is kind of when you add pressure to a solution or pull pressure from the solution. So like if you're getting an injection, that's a positive potential. If we're talking about like the cell wall is going to push back, and so that's also positive potential. The solute potential has to do with the solutes that are in the solution and how that leads to the pressure within it. Okay, and so when you're doing these calculations, there is a solute potential equation. This is the one that's on your formula sheet. So you will have this on your formula sheet. In case you haven't checked out Blue Book, please make sure you check out Blue Book. You can see how the formula sheet kind of drops down. Ooh, Miss Clinton is in here too. She used to always be like my bestie in the chat. Should I just make her an admin so she can do stuff? Let's see. Make moderator. Where are you at? Oh, I can't figure out why I can't add her. Oh, I'm sorry. Anyways, so McClendon is here if you want to ask her questions too. She's all there. Okay, anyways. So the ionization constant, this has to do with that this is how many ions we're going to have to associate within the solution. So like sodium chloride makes 2 ions versus if you're talking about like sucrose, there's 1 because it's a covalent bond. It's not going to kind of dissociate in water. Okay. So concentration is just the concentration of the solution. R is going to be a given value that we have, and it's a constant. So this is, of course, given to you on your formula sheet. And then, of course, our temperature is going to be our degree Celsius plus 273 to get it into Kelvin for us. Okay. Why are you not clicking? So somebody says, do the calculation. So this is from my Math Monday. So if you're having trouble with math questions, if you go to my website and you go to Math Mondays, I have every single formula on the formula sheet, solved out for you with example problems. And then I have like another problem to help you out. So here was one of the example problems. So, we said that we've got potato cores. They're in varying concentrations. They didn't gain or lose mass when they're at 0.32 molar. Use the information to calculate the solute potential, and they tell you the temperature is 22 degrees Celsius. Okay. So since I'm in sucrose, that tells me that I have an I of 1 because the fact that it's not going to make any ions. The concentration is 0.32, which was given to me in the prompt. Okay. I'm given my R, of course, that's just part of the equation, and then I'm going to do 22 plus 273, which gives me my 295, which is my Kelvin, okay? So then if you sub that into the equation, so negative I, C, R, T, that gives you negative 7.84 bars, okay? And so this is the units that was on that last slide that I clearly did not give you. So back to unit 1. So how do we find the negative I value in the equation of negative ICRT? It's the number of ions, okay? So as we talked about with ionic bonds, it just has to do with if the electron has been stolen by another one. So like Na plus and Cl minus. So the way that they're together is that they're attracted. The positive and the negative attracts to each other, and because of that attraction, that creates this like compound, right? And so when the water gets in there, the water has a partial positive and a partial negative, right? So the partial positive is attracted to the negative chlorine, and the partial negative is attracted to the positively charged sodium, which then causes it to dissociate and move apart. Okay, so our sodium chloride, NaCl, will be an I of 2 because it makes two ions, sodium and chlorine. Okay, sucrose is only going to be 1 because of the covalent bond, and we're not going to see that the water is able to dissociate it to separate them. Okay, so next question, explain the protein folding structures, especially the tertiary and the quaternary. Okay, so our primary structure is just a string of amino acids, a long strand. As we said, it's the N terminus to the C terminus. Okay, and so the bonding you'll see between them is, of course, between the carboxyl group and the amino group. Okay, that makes that peptide bond. And so we need to make sure we know primary is just this long string of amino acids. Okay, secondaries are alpha helix and our beta pleated sheet. Okay, so that makes that alpha helix, beta pleat. This is due to hydrogen bonds in our backbone. So the carboxyl group of one amino acid and the amine group, so the hydrogen of the amine group upstream somewhere are going to then be able to then create this hydrogen bond, this attractive bond, which then creates that alpha helix or that beta pleated sheet. Okay. Somebody says solute potential is always negative because the solute molecules are attracted to water molecules and lower the ability. Yes. So we'll look at a picture in a little bit later about that shows us about the free water versus the non-free water. Anyways, so tertiary is our three-dimensional structure. It's the final kind of big thing that you see. Okay. And then this has to do with any bond in the R group. So we could see like hydrogen bonds in our R groups. We can see ionic bonds in the R group, covalent bonds like sulfur, the disulfide linkage type of covalent bonds. You can see the sulfur is kind of creating a bond together. That's what makes your hair curly, by the way. Anyways, you could have hydrophobic interaction where all the hydrophobic R groups are going to like kind of come together in the middle. So you have any type of bonding, any type of interaction that you've seen. Van der Waals, if you want to throw that in from back in chemistry, you could throw that in there, okay? Quaternary is if I have multiple polypeptides. So like hemoglobin is multiple polypeptides, so you kind of have them together, okay? And again, they're just bonding with their R groups. There are no practice exams that are in Blue Book. Those are all SAT practice questions, but if like practice exams. But if you go to the practice questions button, there are practice questions on there. I think it's like 12 questions. And then you can see the answer key for those if you go to the CED. The CED at the very end has an answer key, and it also has explanations for them all. So if you're like, well, I can't check my answers, the answers are all in the CED, so you can definitely check those afterward. Okay. So here shows you the different kind of visual of what I was talking about with the folding. So here would be our primary structure. Here's that secondary structure, looking at alpha helix, and tertiary would be when we get the final three-dimensional shape. And then quaternary is our multiple subunits. So as we said with hemoglobin, it is quaternary. There are two alphas and two betas that make it up. Okay, so are there any key topics we need to know about macromolecules? Yeah, this whole unit, like the unit 1 was mostly about the macromolecules. So definitely know what elements are included in them. So which ones have the nitrogen, which ones have the phosphorus, okay? I find it easier to figure out what certain ones specifically have than trying to remember every single one of them. You could probably also use the amoeba sisters, what is it, the chop, whatever it is. Anyways, so there's certain ones you need to know. Like phosphorus is going to be found in your nucleic acids as well as you'll find in like your phospholipids. Nitrogen will be found in nucleic acids as well as your proteins. Sulfur will be found in our proteins. And so there's certain ones you'll find in these different ones, okay? So I do have my little like slide. So this was from like the unit 1 video where I went through them all, okay? And so you can kind of see like what is it made up of, what is the monomer, know the general structure of them. So as I said, the peptide bond is between the carboxyl group and the amine group. It goes from N terminus to C terminus. And then here is again a reminder of all those structures, and then the way that we're going to see the folding of those R groups. I don't know when the next CRAM session is going to be. I have to figure that out because there's too much happening in my life. Nucleic acids, again, we can see the different elements that make it up, the monomer for it, the general structure for it. Remembering that DNA is going to be anti-parallel. Okay, so we know that we have the directionality of the 5' of the phosphate group to the 3' of the hydroxyl group. That's part of that sugar. Knowing the bonding of it, of adenine and thymine, cytosine and guanine. Knowing about that DNA has a thymine while RNA has a uracil in it. Knowing the sugar and how the the DNA is going to have deoxyribose versus the RNA is going to have ribose, knowing about the strandedness, that DNA is double versus RNA is single stranded. So kind of knowing the difference between purine and pyrimidine. Purine is going to be your double ring, versus pyrimidine is your single ring. You know that you have pure silver, Ag, and you want to cut the pyramid. So kind of remembering all of that. Lipids, there are three different types. You've got your fats, your phospholipids, and your steroids. The big thing about these is that they're all nonpolar because of the fact they have all these carbon hydrogen bonds. As we said, the saturated would be all these single bonds, and the unsaturated is at least one double bond. Remember, I had that typo earlier. I'm so sorry about that. And then we have our phospholipids, looking at that. We have a phosphate group instead of one of these fatty acids. We have the phosphate group with the two fatty acids on it, and that makes up your fatty acid tail, phospholipid bilayer. We're going to see that the nonpolar tails will face the interior of the membrane, and then the heads face the kind of the exterior of the cell and the interior of the actual cell. Okay, so steroids are going to be four fused rings, and we usually see them in intracellular reception. Okay, there's some chi-square stuff. I can make sure I can add them in the next little thing. Okay. Carbohydrates, again, you can see they're made up of carbon, hydrogen, and oxygen. The monomers are monosaccharide, and they're probably just going to give you pretty simple things with this. They're not going to ask you examples. They're probably not going to even go that crazy, okay? The difference between starch and cellulose, it has to do with that this is an alpha linkage, this is a beta linkage, in case you're trying to figure that out. Alpha is going to be on the same side, while beta kind of alternates back and forth. And so you see the whole structure has been flipped. So here's the CH2, and then here's the CH2. So it actually flip the structure. And then knowing where we might see these at. So the structural would be like cellulose and chitin, which is going to lead to the structure that we'll see in these different molecules. And then storage would be like the starch and the glycogen. Okay. Whew. So that was all of the Unit 1 questions that were submitted. I'm sure there were way more that were submitted in the chat. I'm so sorry I cannot balance them all. It is already 5:30, and I'm just trying to make sure I get this pretty fast for y'all. So there were some exam questions submitted, so I'm just I have these kind of interspersed in between the units just so that they weren't all crazy at the end. So how do you determine the type of graphs you're going to use? So if it's categorical data, so you've got specific individuals, you've got specific sections of a fish tank, you've got specific sites of a water sample, you got these specific things, and they really don't connect to each other in a linear fashion. Linear would be your line graph. So we're looking at time, 5 minutes, 10 minutes, 15 minutes, 20 minutes, or concentration, 5, 10, 15, 20, or whatever the concentration is. Okay. And then if you have independent data, it's a scatter plot. And you'll see later when we look at an example of this, what I mean by independent data. Okay. So here we have a question, right? They tell us we have the rate of consumption of ATP synthesis. I see that I have rate of oxygen consumption, rate of ATP synthesis, and I've got this data. So do me a favor and go ahead and in the chat, I want you to tell me what kind of graph you think this is. Okay. And while you're doing that, I'm going to see if I can kill some time and answer some questions on there that we have going on in the chat. Okay. So I'm seeing that, 5', 3', yes, the 5' is where your phosphate group is. The 3' is where you're going to see your hydroxyl group of your sugar. And you're going to see that that is the directionality for it. So when we talk about transcription and translation, and we talk about replication, there's a certain directionality that has to take place with those. What is pyruvate? Pyruvate is going to be, after you break down the glucose, you make the pyruvate. Okay, so I'm seeing a lot coming in the chat. I'm seeing some bars, I'm seeing some lines, all this other stuff. So in case you didn't notice it, I also included the question. So the question says, using this template in the space provided, construct a bar graph that represents the data shown in table 1. So y'all, sometimes they will tell you it's a bar graph. They will say, hey, this looks a little obvious. This could be line, this could be bar. So I want to make sure that the students are going to make a bar graph. So they're going to say, construct a bar graph. And they will tell you what kind of graph to make. Okay. Or they'll make it obvious by using certain axes, and they'll tell you what it is. Okay. So make sure that you are looking at your prompt to see what kind of graph to make. If they tell you, do not make the wrong graph. Make sure you are making the correct graph, okay? You will probably not have to calculate standard deviation on the exam. That's going to take way too long, but they may give you these error bars that you would have to graph on there, okay? Your x-axis would be your temperatures. And your y-axis would be your rate of metabolic marker, and then you would have 6 bars on it. You would have the bars that show your oxygen consumption and the bars for your ATP synthesis. Also, when I'm thinking about it, just mentioning, sometimes y'all like to have multiple colors. You have a blue pen and a black pen, okay? The scans are all in the same color. We do not see the difference between your black ink and your blue ink. So if you're like, oh, the blue ink is my oxygen, the black ink is my ATP, y'all, the readers can't tell that, so please don't do that. And make sure, like, so maybe like hash mark one of them, shade one slightly. Don't like make a like long time of shading, but like shade one of them. And when you're doing those error bars, make sure that you do not cover up the bottom of that error bar with your shading or whatever you choose to do. Okay. This was actually from the 2024 exam, if I remember correctly. So that way, like, you would be able to kind of go back to if you need to. And in case you're trying to like figure out FRQs, I have a thing called FRQ Friday where I've gone through every single FRQ from 2013 to current, every single one of them, in which I go through and I've explained every single one of them to you. P.S. In case you're wondering, I'm just killing time while I try to get you to answer this next one. What kind of graph do you think this is? Yeah, the readers cannot see colors. It is a scan. It's a black and white scan. If you go to AP Central and you look at like the sample responses, you can see what the graphs look like for your readers, and you can tell that all we can see is the black. So we really can't see any of the other colors on it. There could be math. Yeah, there's definitely gonna be math on the graphing. I've been doing math problems every single day since whenever I started my review, just showing you different types of them. Okay, so I'm seeing some bars coming in the chat. As you notice, I didn't give you the question because the question specifically didn't actually tell them what kind of graph to make. Last time I was just seeing if you could, follow it. So here we're seeing that I have 6 different species. And so you're thinking to yourself, well, Ms. Jones, you said if I had individualized data, right, that must be something, right? But in this one, they wanted you just to make a bar graph. We were looking at the difference between the like the species with like the normal and the elevated, okay? So we were able to actually make this into a bar graph. I did not want this to be a scatter plot. You did not get credit if it was a scatter. It had to be a bar graph for this one, okay? And then here's our next one that we have, okay? How did we differentiate different lines? Different lines, you could label the back of them. So at the end of the line, you can say what sample it is. You could also make one of them like a hash mark versus one of them a solid mark. So there's a lot of different ways you can differentiate your lines. Symbols could work along a line if you want to use different symbols. I would probably just do a straight line versus like a hash line, just because of the fact that like it's hard to figure out, okay? So this next one, as you clearly see, I've given you the question. I'm waiting for y'all to answer in the chat, but no one is giving me anything. You just keep asking more questions. So I'll just go ahead and tell you the answer. So on this one, I kind of didn't give you all the information that you would need. They gave them on one axis the number of double strand breaks, and on the other axis, they said the average of a crossover. So they went ahead and labeled their axes for them. And so they were looking for a connection between the double strand breaks and the crossing overs. They were looking for a correlation between that. Okay. And so this one, since I wasn't looking for a correlation between normal and elevated, I was just comparing normal and elevated. That's why that one was a bar graph. But since I'm looking at specific different strains, and I'm looking at the orientation of this one with this one, those are independent data points, or independent. Strain 1 has nothing to do with strain 2. And so because of that, and I'm looking at the relationship between this to this, that's going to be a scatter plot. And I'm seeing a lot of bar graphs coming in. And so it is sometimes hard to tell. But so just do your best on the exam, no matter what happens. Even if you make the wrong graph, okay, that's only going to be 1 point on it, okay? So there we go. So on to unit 2. Oh my God, it's already 5:37. We are doomed, y'all. So what things can and cannot cross the membrane? So in terms of your membrane is nonpolar. Okay, we said that it's made up of a phospholipid bilayer. So you have these 2 phospholipids that are kind of facing each other. So the interior part of it is going to be nonpolar, and the exterior part is going to be hydrophilic, and it's going to be polar. Okay, so anything that is small and nonpolar can get straight through that membrane without having any type of issues. It goes right through the membrane. Okay, versus if it's charged, if it's polar, it's not going to be able to get through. And so that's when we have facilitated. Facilitated has to do with the fact that we're looking at something that is small and it's polar or it's charged. And so it can go through a transport protein, so a carrier protein or a channel protein, okay? Now, both of these that I'm talking about are both talking about passive transport, things that are moving straight through without needing any type of energy added to them, okay? Versus if we think about like active transport, that's going to apply some type of energy, and it's usually going to go against the concentration gradient. We'll still use a transport protein to do that, but this was talking about like what can and cannot. Usually it's going to go down membrane, and it's usually going to need something to get across the membrane. If it's polar or charged, if it's non-polar, it can just go straight through the membrane, okay? So here you can kind of see that we're looking at simple diffusion going straight through, doesn't require any type of protein, versus here we see our channel protein or a carrier protein. Channel protein is just like a little like basically a hole that you have in the membrane. Not really a hole, but it's protein that surrounds like a channel, a little tunnel, and the materials can just pass straight through them. Hydrophobic is to be nonpolar. Hydrophilic is polar, yes. And then here, if we're looking at a carrier protein, it's going to bind, change shape. Anytime you bind something to a protein, it always changes shape. That's something you need to make sure that you know. If you're talking about any enzyme, any protein, anything out there, and you say that this thing bound to this protein, the answer is always going to be that it changed shape. Now, the change of shape then leads to potential other questions, other answers, but you always have to talk about the fact that it has changed shape. Okay, so how does that relate to cell communication? So in terms of cell communication, if you have a protein hormones are going to be polar because of the protein. Okay, and we know that proteins are going to fold to their their hydrophilic R groups are outside. So the protein itself is going to be polar. Okay, so that means the receptor in cell signaling is going to be on that membrane. Okay, so it's going to be an extracellular receptor. It's going to be sitting on the membrane, and then it's going to lead to the signal transduction from there. Versus steroid hormone is going to be an intracellular receptor. Okay, there's a receptor on the inside of the cell. Well, how is it going to get there? Well, if it's a steroid, we talked about steroids are nonpolar. The steroid can get straight through that membrane without any type. It just goes straight, simple diffusion, straight through. Okay, and then it's going to bind to a receptor that's on the inside of the cell. Okay, and so that's how it kind of relates to cell communication. So then someone says, well, can you talk about osmolarity? Osmolarity is just the measure of the solute concentration. So you can be hypoosmotic, which means that there's a low solute concentration, or hyperosmotic, which means there's a high solute concentration. But I don't think that's what the person was trying to ask me. I think they were trying to get to tonicity and talking about like, what is the tonicity, hypertonic, hypotonic? I think that's what the student was actually trying to ask. So we're going to go to this. So earlier I mentioned that there was a diagram I really like. This is the diagram I absolutely love, and this has to go with that solute potential. Okay, and so here we see that this one has no solutes in it. And so because of the fact that there's no solutes in here, okay, this water is free. There's a lot more free water. It's not bound to anything. It's not restricted. So this is where we see that we have a very high amount of free water. Whereas over here, while I have these solutes, each of these water molecules are going to have to surround that solute. Okay, and that's kind of how things dissolve, is that the water surrounds each of these different particles. Okay. And so, no, I don't have the math built into this for the cell size ratio math. But if you go to my Math Monday, I've got the math done for every single formula on the formula sheet. So here we see, of course, that solute is in there. We surround it. And so there's less free water. And so because of that, there's less free water, we see that the water is going to move from where there's more free water to where there's less free water. And so tonicity is just the ability for a solution to see the movement of that water across the membrane. Okay. So hypotonic means there's a low solute and a high free water. So the water is going to move out of a hypotonic solution. Hypertonic means I have a high solute and a low amount of free water. So the water is going to move into this. The goal is that we're trying to balance. Okay. Now, the thing you need to understand is that if one solution is hypertonic, the solutions on the other side of the membrane is hypertonic. Okay. They're going to be opposite of each other. If you stand next to somebody and you're taller than them, then they're shorter than you. So if one's hypo, the other one's hyper. Okay. And the water's going to move from the hypotonic to the hypertonic. Okay. Through that membrane from one to the other. Okay. Now, water is going to require a channel in order to move. It usually has an aquaporin, which is the channel protein. But it is able to leak through. So if you ever see a question that's talking about water leaking through the membrane, it's going to move through the membrane. I know it's small, I know it's polar, and it will go, but it's not as efficient as if it was to go through it. Somebody says, remember, solutes suck. So if there's a high amount of solutes, it's going to suck the water. It's going to pull the water into that environment. So thank you for that, Janine. And then isotonic means that we have a equal amount of solute, so the equal amount of free water on both sides. Okay, so that means that there's going to be the same amount of water moving in as out of that solution. It's just going to kind of be an equilibrium and going back and forth. Okay, so in terms of the hypo and hypertonic, here we would see our cells that are in a hypertonic solution. So the solution is hypertonic. Okay, so the water is going to move from this hypotonic cell to the solution. Okay, isotonic, we're going to see the same amount of water going in and out. And then hypotonic solution, the water is going to rush in, which then causes the cells to burst. Okay. And so this isotonic is where the cells kind of want to be. Yeah, I know. Every year someone tells me that mnemonic, and every year I forget it. So the isotonic is where we're going to kind of want our animal cells to be, because that's where we're going to have equal amounts going in and out. We don't have a cell wall to kind of protect us, but a plant does have a cell wall. Okay. So because of that, they prefer to be in this state right here with a hypotonic solution. They prefer to be turgid, okay? So this thing right you're looking at right here is a central vacuole. So the water goes into that central vacuole and it creates a pressure, okay? That pressure then kind of pushes against the cell wall that inhibits any more water from coming in, okay? It kind of makes it nice and firm. Versus isotonic is going to be flaccid and kind of limp. And then the hypertonic solution, we're going to see that the water moves out of the cell, which then causes plasmolysis. So the cell membrane pulls away from the cell wall. And so it kind of gets like squishy. So do we need to know about uniporters, symporters, and antiporters? I don't think they're going to use the words uniporter, symporter, and antiporter. They might, but I don't think they will. So if you see this and you're like, I don't know what that means, let's break this down and let's think about things that we do know. Uniport. Okay, so uni makes us think of one. So that must mean that if it's a, port, you go to like cruises, you've been on ports before and you're like, that's where you stop, that's where you move things across. Okay, cool. So this means that we must be binding and transporting one substance because it's a uni, right? Okay. So symporter makes us think about like, okay, well, I've got sim. Sim means together. So like you think about like symbiosis, they're together. You're thinking like, yeah, okay, whatever. So sim makes us think of together. So these materials are going to move together. So we have like two things moving through that transport protein together. Okay. And then antiporter means that they're going to be moving opposite. Sodium potassium pump is the example of this. Sodium is going to move one way while potassium moves the other. So those are going to be antiporter. They're going to move in other ways. Okay. So what would they be called other ways? I don't really know what else they would call them, but I really just think they're going to say this is a sodium channel, or they may say like this channel requires protons and potassium. So like there was a question about the morning glory flower a couple years ago that talked about that you needed both of them to move at the same time. And so because of that, you had a diagram that showed you it. Like you weren't probably gonna need to have to like add this too. So what's affinity? Affinity just has to do with the attraction, so the degree of which a substance tends to combine with each other. I think that you might have been thinking about like electron affinity. I'm not really sure where that was coming from, or like, I don't know, but it just means that they're attracted in some way. So see, I'm telling y'all, like something happened with my slides. They got all out of order. I had a picture. So here's the uniport. It's going to transport us again a single substance. Symporter has to do with two substances moving together through it. And then the antiport has one going in and the other one coming out. So like they're kind of like in opposite directions. And how many times this like messes up? Anyways, can you explain the sodium potassium pump? Okay, so since it's a pump, we know that we're having some type of energy going into it, right? So this means that we're going to be going against our concentration gradient. And so with the sodium potassium pump, we're going to see that we're going to have our sodium is going to bind to it and it's going to pump those out because we see that this phosphate is going to go through hydrolysis, right? And it's going to phosphorylate that material. If it's a protein and you bind something to it, it always changes shape. And so it changes shape, which then moves the sodium to the other side. It releases it. And then our potassiums are going to bind. And then when that phosphate pops off, it then changes shape and then moves it back. So the way that I remember the directionality of this is that if Nana's at your house, she's been there for too long, you don't want Nana there, you kick Nana out. And some people like Na is Grana, like Nana, like, oh, okay, sodium. So we kick sodium out. And then your kids are out partying all day, so you want to drag your kids back in, and K being your potassium. So that's the best way I could come up with to try to help you out there. Okay, so what are the main components of the cell? So big things of our cell, okay, is that we're going to have our phospholipids, of course, has the phosphate group and the fatty acid tails. They're going to be a bilayer, so they face one another, and they're kind of interior, okay? We also have glycolipids. These are going to be for cell communication, and we also have glycoproteins, the same thing. Just the glycolipid is a sugar attached to a lipid, and a glycoprotein is a sugar attached to a protein. You also have transport proteins, or you could just have peripheral proteins. You just have all these proteins that sit in the membrane. So like your membrane is made up of like phospholipids and proteins, like the big component of it. But you also gonna have like this extracellular matrix, this kind of part on the outside that kind of helps to give rigidity. Since we don't have a cell wall to animal cells, it kind of gives structure and rigidity. The cholesterol's job is to maintain the fluidity of the membrane. And so I was thinking like kids in a marriage, right? So like with the cholesterol, it's going to stop the parents from getting too close together. But then also like if they try to split up, they try to, get divorced, they try to leave each other, they kind of, the kids hold them together and kind of hold the family together. So I don't know. Some ways it helps, sometimes it doesn't. I don't know. Anyway, so here you can kind of see the different components of it. There's just some anchored membranes. There's like transport proteins. There's just all types of membranes. Glycolipid versus glycoprotein. Again, they're just for cell communication. So the glycolipid is going to be the sugar attached to a lipid. Glycoprotein is the sugar attached to a protein. Did I say that right? Sugar, lipid, sugar, protein. And so the glyco makes you think of sugar. And if you remember like glycolysis, you can remember sugar. So that helps. I don't know. So explain the difference between outer and inner membranes for the mitochondria and the chloroplast in terms of their functions. Okay. So here we have the outer membrane of the mitochondria, and then here we have the cristae, and then here we have the outer membrane of the chloroplast. There's an inner membrane, but I think they're asking about this thylakoid. Okay. So in terms of our mitochondria, okay, the outer membrane is just like surrounding the organelle. It's like keeping the organelle its own little compartment. It's separating it from the rest of the cell. Okay. And then as we go into the inner membrane, right, this is the cristae, which is that inner membrane. And its job is for the oxidative phosphorylation. So this is where we're going to see oxidative phosphorylation in Unit 3 when we talk about like cell respiration, which I don't think anyone asked any cell respiration questions. Whatever. So that's where we're going to see the electron transport chain taking place, which is going to pump those protons. It pumps the protons into this ion space. So the spaces between the outer membrane and the inner membrane is where we're going to see that high concentration of our protons, okay? And then the matrix is going to be the site of our Krebs cycle. So on the inside of that cristae, on the inside of the inner membrane right here, we're going to have that matrix, which is where the Krebs cycle will take place. So I think I said everything that's on that one. So then we talk about our chloroplast. So we have our site of our light reaction. So the little thing right here, these are the thylakoids. And so in there are the components of the light reactions. And so we'll see that that's where we'll see the electron transport chain taking place for those light reactions. And so the stroma is just a jelly substance on the outside. Remembering that the chloroplast and mitochondria are prokaryotes. And so this is just the cytosol of this prokaryote of that. We call it the stroma. So the stroma is where we see the Calvin cycle versus the thylakoid space is where we're going to pump those protons into the inside of this little thylakoid. And that's where we'll see that the pH is going to go down because of the fact that we have an increase of our H pluses. And if you remember, those are going to be inverse of each other. So if you increase H plus, you decrease the pH. So I think I've said all that. Cool. Explain the difference between endocytosis, phagocytosis, and exocytosis. Phagocytosis is a type of endocytosis. So let's first look at endocytosis versus exocytosis. Endocytosis is going to be bulk movement into the cell. So we're going to bring something into our cell. If you need a way to remember that, endo and enter, start with the same little letters right there. Exocytosis has to do with bulk movement out of the cell. So we're moving things out, again, exiting the cell. So we're moving things out, okay? So here we can see that phagocytosis is going to be cellular eating, okay? So these little pseudopods are going to surround the food. You see this in our macrophages, the white blood cells within your body. They're going to go and surround this bacteria, and they're going to bring it in. It's then going to form a food vacuole that will fuse with your lysosome so they can digest and break down that bacteria, that food, that whatever was consumed. Penocytosis is going to be cellular drinking, okay? So it's going to bring in solute, and as it's trying to bring in this extracellular fluid, sometimes these solutes are dissolved in that fluid, and so it's kind of like drinking and bringing these solutes in, okay? And then receptor-mediated endocytosis, we have signaling molecules, these little stars. They bind to the receptor, and it causes it to go through like this invagination. It makes a little pit, and then it forms a vesicle to bring in some specific material for it. This is just part of signal transduction, okay? Exocytosis is things moving out, okay? So we've seen before on exam where they talk about like what is the path that things are going to take. Well, of course, our proteins are going to be synthesized in our rough ER. They then go to the Golgi to be packaged and modified. They then go through their vesicle, and then they come and they fuse with the plasma membrane and go through exocytosis and release it, okay? So that's kind of where you need to be focusing on with the exocytosis is how is it getting out. Yes, go walk your dogs. So is it possible I can help you understand water potential more? Because, watching the video is so confused. Okay, so water potential is just the potential for water to move. Like, how is water going to move? It's always going to go from high to low. Everything moves from high to low, okay? So we're going to move from a high water potential to a low water potential. So over here, we see that I have a water potential of 0. And here I have a water potential of some negative number because of the fact that I've got this solute in it, okay? You cannot wear your Apple Watch on the exam because of the fact that it is able to receive messages. You can't have your Fitbit. You can't have your, any type of smartwatches. Like, it's like no tech. Like, but there's a timer on your screen, so you don't need to worry about needing a timer, okay? So someone says, can I relate water potential to a plant or animal, something they can follow, okay? So here we see the water potential of the soil is negative 0.3. And then here I see like up at the xylem is negative 0.8, okay? That's going from a high water potential to a low water potential. And you may be thinking to yourself, Ms. Jones, 0.8 is definitely greater than 0.3. but not negative. Okay, look at it on a number line. You'll see that -8 is here, -3 is here. So you see that this is higher than that. Okay. Anyways, so that kind of shows you like how water can move up. Yes, we have cohesion and adhesion. Cohesion, adhesion that's allowing the water to move up, but it's also we have root pressure and we've got all these other like kind of water potential that might be helping to move it up too. So hypotonic solution versus the hypotonic cell. Well, the hypotonic solution is talking about the solution itself is hypotonic. So the solution has a low solute and a high amount of free water. Versus a hypotonic cell, the cytosol for it. My camera is covering part of the question. I'm fine. You know, y'all, I tried here. Okay. The cytosol is hypotonic. So the outside of the environment is hypertonic, and so the water is going to move out of these cells. So whenever you're writing an FRQ question, you talk about something being hypo or hypertonic. Can you please make sure that you talk about with respect to? The cell is hypotonic to the environment. The environment is hypertonic to the cell. Give it a like kind of difference between them. You can't kind of go through it. Yes, the slides will be posted later. I'm telling you, I finished this 10 minutes before I came live. So proteins are made in the rough ER, lipids are made in the smooth ER, where the carbohydrates and nucleic acid get made. So I had to look up this part, the carbohydrates, because like we really don't talk about it much. But the carbohydrates are made in the mitochondria as part of whatever. It's part of the conversion of the pyruvate to the phosphoenediol pyruvate. And then the phosphoreticulatum also kind of works a little bit with the glycoproteins, the glycolipids. Okay, I really don't think you're going to be required to know that, but the person asked the question, and I was like, hey, let me look this up. Okay, so nucleic acids, yeah, it's in the nucleus, right? We go through replication in the nucleus. You may not have your phone in the 10-minute break. Your phones will be locked away, children. Nucleoids. Has to do with our prokaryotes. Okay, so there's just gonna be a certain region that we're gonna have inside of the cell. Because these will be our prokaryotes, so they don't really have that nuclear envelope. But wherever the DNA is where we're going to see the nucleic acids getting made. Okay, and as a reminder, there are nucleic acids inside of your mitochondria and your chloroplast. Okay, so whenever you're trying to talk about like where is the DNA in the cell, there's some in the mitochondria, there's some in the chloroplast, and there's some in the nucleus. Okay, so don't forget that there is DNA inside of your mitochondria and chloroplast, because students kind of forget about them being in prokaryotes. Yes, they will take your phone or they will tell you to put your phone away. My school, they're going to be putting them in their backpacks, and their backpacks will be locked away in a closet. So they will not have access to their phones or any of their electronics. And the same thing as you should not be able to use your phone. That allows you to potentially look things up at the break, which is not appropriate. Okay, so surface area volume ratio, which should be higher. Okay, so for your cell to be most efficient, you're going to see that it's going to have a high surface area to volume ratio, which means that I've got to have a high amount of surface area in comparison to a low amount of volume, right? That gives you a high ratio between the two numbers. You really don't need to store any formulas on your graphing calculator because they give you the formulas on the formula sheet. There's a little drop down about it, okay? And you probably should not be talking to people during the break. I don't know what the directions say, but you shouldn't really be talking to anybody. You should just be quietly eating your snack, getting your brain wrapped around that, hey, I gotta go get these FRQs done. So what are peroxisomes? These are just oxidative, and these contain oxidative enzymes, and their job is to kind of help to detox and break things down using oxygen. And so they use molecular oxygen to remove the hydrogen, and they form hydrogen peroxide, which is why they talk about it being peroxisomes, like they're going to make peroxide, hydrogen peroxide. Yeah, I hope you get it. So that was the end of Unit 2. Those were all the questions that were submitted. I did my best, y'all. So there was a question that said, how do I determine the control group in an experiment? Okay. So negative control means that there was no treatment added to it. Okay. So when I'm talking about the experiment and I mentioned how there's this certain experiment and like I'm testing, I don't know, prolactin levels, right? With exercise. So the negative control is going to be the one that didn't exercise, but I still tested their prolactin level. So I'm seeing how does the exercise, how does my treatment affect the product, right? And so I need a negative control to be one that does not get the treatment, so I'd be able to compare it, okay? So whenever they ask, like, what was the purpose of a control, you're not just going to say to compare. Compare what? To determine what? I'm using this as a control to compare with the treatment group to determine, did the exercise have an effect on prolactin levels? Spell it out, because we all know what a control does. We all know it's a comparator, okay? But we need to make sure that we are adding more information in there so that they know that we're talking about that specific question. Anyway, so positive control has an expected result We know, yes, you will find this on YouTube later, I promise. Sorry, y'all. So the positive control is an expected result. You know what will happen. You know that if you do not give this bacteria the plasmid that allows it to be antibiotic resistant, you know it's going to die in the presence of the antibiotic. You know, like if I give it the if I give it the plasmid and I grow it not in the presence of an antibiotic, it should grow. But if something happened and it died, then I know that something happened in my experiment and I can't trust my data. So the point of the positive control is so that you know that your data is valid, okay? That this thing should have happened and it did happen. So I know that my results are going to show me what's happening, okay? Yes, so there's two different types, okay? So here's the question from last year's exam. I'm not reading through the whole thing. I'm just gonna look at the chart because I'm lazy, okay? So they say, like, what is the control group of this? Well, the control group is the one that has none. I don't have the CTF and I don't have the IML, okay? So you could say, like, the none group. I think they actually say up here, like, the group 1 contained no kinetochore proteins. So I could say group 1. I could say the one that was none. I could say the one that does not have the kinetochore protein. So, like, it's the one that doesn't have the thing because they asked you what was the control group. Okay, so the control is going to be the one that doesn't have it. Okay, they then go on to say that there's a follow-up experiment. They created a modified version of CTF in which the binding protein had to be removed. They compared the frequency of the crossing over of the yeast cells in the presence and the absence of it, and then the presence and the absence of the modified. Okay, and they want to know why are they going to use a modified that's unable to bind? Well, if it can't bind to the DNA, and I believe that the binding to the DNA is the factor, like it has to bind in order for this thing to happen, then I'm able to determine, yes, okay, it determines whether the DNA binding is required in order for us to have crossing over, okay? Because if I made sure that DNA didn't bind and I saw like crossing over didn't take place, then I can prove, okay, yes, I know that this process is going to take place. Talk about the determining whether or not the presence of it is enough to inhibit the crossing over. So like kind of giving them a little bit of thing. And when you're looking at these scoring guidelines, the things that are underlined are the things you need to make sure you're stating in the prompt. Anything with a slash means those were options of things you could have said, okay? But anything underlined in that little area, you need to make sure that you stated. I'm going to go through 4 units in this. So clearly, since I'm only on unit 2, I probably have another hour. Sorry, y'all. So we're looking at that, okay? So then they go on. This is question, I think, 5 or 4. It's later on in the question. Like, this is not the same question in the exam. And no, I'm not going through all 8 units. To investigate whether red blood cells lose the ability to take in the glucose from their environment, they collected red blood cells. They're from age 1 to 7, and then they incubate equal numbers in the dish. They want to know, well, why am I going to use equal numbers? Like, what was the significance of using equal numbers, right? Well, the reason why you're going to use equal numbers is so that I can attribute the differences to their age. If I didn't have equal numbers of them, then whatever results I got from this experiment, I don't know, was it because I had more cells? Was it because something about the cell was different? I don't know. So I have to keep them equal in here, okay? And so by keeping them equal, I was able to compare the results between the different dishes. I can eliminate the number of cells as being a variable, right? Because of the fact. And then, of course, they might affect the results if I have a different number of cells. So those were all possible things you could have put, okay? So that gets us through 2. And hey, look, it's 6 o'clock. Sorry, y'all. You are welcome to leave whenever you need to leave, y'all. I told you I would go through these, and I'm just trying to make sure I give you everything I could. I'm only going to go through Unit 4 because that's when I ran out of time and had to get live. So Unit 3. So can I go over the Calvin cycle? Yeah. So y'all make sure that when you're thinking about these cycles that you're not trying to get like super in depth about it. Like you just need big picture of everything, okay? The units that are most on the exam is 3, 6, and 7. So not 4 is like they're all basically the same equidistance on the exam, anyways. So the function of the Calvin cycle is for us to synthesize organic compounds. Specifically, we're making this G3P. Okay, we're fixing the carbon dioxide in order to make this. Yeah, control is untouched. Well, not always, but yes. So the reactants, we're going to see that we have 3 carbon dioxides coming in. Okay, since I have a G3P, it has 3 carbons, so 3 carbons have to come in for 3 carbons to come back out. I have 6 NADPHs, remembering that the P is for photosynthesis, so we know that we have NADPH in photosynthesis, and then I have 9 ATPs. 6 are made here, and then 3 are made there, or not made, but used in those parts. We make them in the light reactions. Okay. Versus, can I go over the the Krebs cycle? The Krebs cycle is going to be complete oxidation. So I'm going to completely oxidize my glucose. Okay, so the first part of the breaking down took place when I was in glycolysis. We broke down the glucose into the pyruvate, and then that pyruvate went through the the pyruvate oxidation, which then allowed us to lose one of the carbon dioxides, but then we make this acetyl-CoA. So the COA comes into the cycle, and the whole point of this is for me to complete and finish breaking down the glucose, right? Break down that pyruvate, break down the acetyl-CoA. And so we're going to see that at the end of this, we do not have any more of that carbon there, okay? And so knowing that I start with the acetyl-CoA, I know that I have 3 NADHs. I know that I've got the 1 ATP, the 1 FADH2, like all that is what's going into it. And this is just kind of providing that energy, right? This is an error. I'm making 3 NADHs. No, wait. Yeah, y'all, this is an error. See, this is what happens when I'm rushing through to try and make this PowerPoint for y'all. We use 1 acetyl-CoA, and I make 3 NADHs, 1 FADH2, and I make 1 ATP. Like, you can see it's made right here. 1 made. There's 1 NADH made. 1 made. 1 made. Sorry. I promise you, I did my best to make this PowerPoint for y'all. I just rushed. Hey, activation energy. What is activation energy? Activation energy has to do with that we have the amount of energy that's required to get the reaction started. So this right here, this would be an exergonic reaction because I'm releasing energy. So you see that delta G is negative. So that's more of a chemistry thing. But we do have the exergonic and the endergonic in our standards to know them. So exergonic means that it's releasing energy. Endergonic means that it's absorbing energy or taking energy from the environment. Okay. So activation energy is the energy that's required to get this whole thing started. Okay. Okay, so explain the electron transport chain in mitochondria and a chloroplast. Okay, so the big point is that the reason why we're doing the electron transport chain is so that I can get the energy in a very small stepwise function, right? We're not going to all of a sudden release the energy. And by slowly releasing the energy, I'm able to absorb the energy in small little chunks, so I'll be able to do something with it. Think about like a turbine. You're going to see the water is going to drop a little bit at a time, like itty bitty, itty bitty, itty bitty. And as it slowly drops, we're going to be able to absorb a little bit of the energy each time. Okay, so that's what we see happening in both the electron transport chains is that I allow the energy to slowly drop. And as that electron slowly drops in energy, it releases a little bit of energy out in the form of us pumping a proton across the membrane, which then creates this gradient that we can then use all that potential energy. Okay, so here we see it. We've got our NADH and the FADH2 are going to drop off their electrons. Okay, notice that NADH is going to drop off at an earlier cytochrome versus the FADH2 is at a later one. So we see that NADH makes more ATP than FADH2. Okay, at each of these points, we just see that the protons are getting pumped across the membrane. So you see that protons are pumped into the ion space. As we said before about the concentration, as the H plus increases in here, that the pH is going to decrease. So we're going to see that there's a low pH in the intramembrane space, so the space that we have between the two membranes. Okay. And so at the end electron transport chain, which is right here, end electron transport chain, at the end, that's when we use that proton gradient to move down its gradient, right? The protons go through ATP synthase, which then turns it to synthesize the ATP. I use the potential energy of this proton gradient to then synthesize the ATP that's needed, right? The same exact thing is happening in chloroplasts, okay? The only difference is that I just have different cytochromes. I have different things in the middle of it that are allowing for this, but I still have my protons are being pushed in to the thylakoid space, okay? Again, the energy comes up, it then absorbs that energy in the form of a pigment, right? The electron has high energy, it drops off the energy, losing it as it goes in the electron transport chain, and it goes into photosystem 1. And then this time it gets taken by NADPH, remembering the P stands for photosynthesis. I know that is a lie, but it is okay because it helps you remember the lie, okay? And then after the electron transport chain, we see that ATP synthase can synthesize, okay? What are the laws of energy? I think they mean the laws of thermodynamics. So the first law of thermodynamics, yeah, thylakoid space will be acidic because of the fact that you have a high amount of protons in there. So first law of thermodynamics, energy cannot be created or destroyed. Okay, so whenever you talk about energy, energy has to be synthesized. Like if you're gonna say that you synthesize the like ATP, like we absorb the energy from something and I then can use that energy to make something else. Okay, so you can't just say like I made energy. No, you did not. You got energy from somewhere else. Okay. Second law of thermodynamics, this has to do with that every single reaction is going to increase the entropy of the system. So everything is going to create disorder. It's going to create chaos in our system, okay? And so every reaction increases that entropy that we have in that system, okay? So seeing photosynthesis, color absorption on the graph, okay? So this was asked by a lot of people. I did not realize that y'all were having so much trouble with this, so sorry. So absorption just has to do with like how much of that light was absorbed, okay? So if you're looking at something that absorbs light, whatever does not get absorbed gets reflected back to you. So the reason why a plant looks green is because of the fact that it reflected the green light back to your eyes, and your eyes, of course, absorbed that light, and it then created like, oh, that's green. Like that's what it sees, okay? So on the 2000, 13, 14, one of those exams, they gave you this diagram and they asked you what was the color of this organism. Okay. And so they gave you a little bit of background. They told you that there was the Bacterodopsin, which is purple, and they had chlorophyll, which was green. Okay. So they gave you background information. You were able to then figure out, like, okay, from this, how can I figure out what is the color of this? Like, which of the two organisms is this an absorbance of? Okay. So if I look here, I have a high amount of absorbance right here in this range of this, like, I don't know, 550 to 600. There's a high amount of energy that's absorbed, right? But where is it not absorbed? It's going to be out here on this outer range. And that's what's getting it reflected. And that's what your, of course, eyes are going to see to be able to determine, oh, what color is this? So if you put blue and red together, you get purple, which is the Bacterodopsin, like, because they told you that the Bacterodopsin was purple. And so you were able to figure out that this was purple because of the fact that it reflected the blue and the red that make purple. Okay. This one, they ask you, well, what color is it? Well, logically, if you figure out the Bacterodopsin was purple, you could, of course, figure out what color this one is. So we can see that we have a low amount of absorbency here, which is green. So this is our chlorophyll because of the fact that it reflected all that light. And so another part of that question was asking about like the rate of photosynthesis. So we have a high amount of rate of photosynthesis here, right? At the 430 area, we have a high amount of photosynthesis because it has the most amount of energy. And the more energy that you have, the more photosynthesis you can go through. Then this one over here at like 650 was like the next highest. And then like the 550 was the lowest rate of photosynthesis. And that was like, honestly, that was 5 points on the exam. Like, it was a lot of points if you just like identified and then like explained a couple things from the diagram. So a lot of points can be made from just looking at a diagram. Okay, so another question from 3. Can you explain what we've learned given evidence of chemosynthetic organisms? So the chemosynthetic theory just talks about that life originated through a series of combinations of chemical reactions. Thank you, Stanley Miller and Handel. Yay. Anyway, so how can one enzyme bind to multiple different substrates? Because they have multiple active sites. Okay, so it's just a protein. A protein is just a chemical. It's folded, and there's all these little spots on it. And if there's anywhere on that something else can bind to it, then that's going to be a site, some type of allosteric site. Okay, and so because of the fact that it's that site on it, that it can bind. Okay, so when we're looking at like our enzymes, there's a specific active site in order for that enzyme to do its job, but also has these allosteric sites that where it can bind. Okay, and if something else binds to that site, if you bind something to a protein, it always changes shape. And so like here we see with this non-competitive inhibitor, it bound to another site and then it changed the shape of the active site. So now my substrate doesn't bind anymore. Versus like a competitive inhibitor binds to the exact same active site because it has a similar shape to it. So do you remember all the small processes that take place in the Calvin cycle and what goes in, what goes out? Well, yeah, just know big picture, okay? Why did we do this? So what goes into it? What happens? What goes into each of the processes? What comes out of the processes? Where does it take place? And why is it important? Okay. So we went through a couple of those. If you need to see that again, I have a TikTok that goes through all of them really quickly. So if you go to my page, you go down to the TikToks button, you're going to see like a spot that just says like, I don't know, cell respiration overview, and you can see all of them in like a 1 minute video. And I go through them all. I make a like beautiful little chart for you. I forgot to get the chart added, so that's why I'm just telling you where the chart is. Okay. So do you need to know the names of the enzymes in cell respiration photosynthesis? Nope. Do not know the names. You do not need to know all the little nitty gritty, teeny tiny little details. You just need to have the big picture. Chlorophyll gets broken down and those nutrients are reabsorbed by the tree until spring. Yeah, so that's why you see it is because in the fall, the chlorophyll is no longer being synthesized, and so you're seeing the oh gosh, what are they called? They're the chromatones. It's from the other organelles. My brain is currently going blank of what those antenna pigments are called. But what's happening is you're seeing the other pigments that are in the plant and then the leaf. And so you're able to then, of course, see the oranges and the yellows and all the other colors that are taking place. Anyway, so like this. So in the past, there was a question. So if we had a question, I think that was on like glycolysis. And they showed you the steps of glycolysis. And they also gave you where a certain enzyme works. And then they said, if this enzyme was inhibited, what would happen? But they told you what the enzyme did. They told you where it worked in the process. So you really didn't have to know that enzyme. It's going to be an application question. They're going to then, you know, if this enzyme is inhibited, what will happen? Like they give you everything you need. You just have to apply the information. Okay. So what is chemiosmosis? Actually, I just wrote chemiosmosis. I don't really know why this was a question, but whatever. Okay. So this is just ions moving across the membrane down electrochemical gradient. So like protons moving from the ion space to the ATP synthase is back to the matrix. That's chemiosmosis. The protons moving back from the thylakoid space to the ATP synthase through the stroma or to the stroma. That's also chemiosmosis. So like when we talk about like oxidative phosphorylation, it's made up of ETC and chemiosmosis. Okay. So explain fermentation. Fermentation is what takes place after glycolysis. Okay, so we have glycolysis. Glycolysis goes through. We make that ATP and we make these NADHs, right? But here's the problem: if you have no oxygen to be the final electron acceptor, then you're not going to be able to remake the NAD plus. And if you have no NAD plus, you're not going to be able to continue to break down glucose. And so if you can't break down glucose, I don't know what's happening. So you can't actually make any energy. You can't make any ATP from that. You can't break down the glucose to get the energy to make the ATP, whatever. Okay. So the whole point of fermentation is so I can regenerate the NAD plus. So I allow and this is lactic acid fermentation. That pyruvate becomes lactate. And then by allowing that, we can then, of course, drop off that electron. So I now have NAD plus that can go back over here to glycolysis. Okay. Everything can do glycolysis. Animals, plants, prokaryotes, we all have the ability to do glycolysis. And so we traditionally see fermentation taking place in these yeasts and these bacteria and whatnot, because of the fact that they don't have a mitochondria. But yeah, anyway. So they asked about CAM plants and C4 plants. This keeps coming up. I don't know why y'all keep asking about it. CAM and C4 is not in the curriculum. It's not required to know about CAM and C4. It is an illustrative example, okay? Which means that if they ask you a question about CAM and C4, it has to be given to you in some type of prompt. They have to tell you about it, right? So the whole point of this is a modification to decrease photoaspiration. It is an evolutionary adaptation, okay? So these organisms that live in this area, they're going to be found in places where it's very hot and arid, and so they're losing their like water through transpiration. And we all know that you need water for photosynthesis because the water is going to provide the electrons needed for photosystem 2 whenever it, of course, goes to electron transport chain. Okay, so if they close their stomata, they close that pore that's in their leaf, they're not bringing in carbon dioxide. They're not going to, like, yeah, so that's a problem because they don't get carbon dioxide, but if they have it open, then they lose all their water, which is also a problem. So what happens is the C4, the spatial separation, there's a different part of it that's going to fix the carbon dioxide. So it fixes the carbon dioxide, it makes oxaloacetate. Those of you like, Miss Jones, I've seen that word before. Yeah, that's in the Krebs cycle, don't worry. So they make the oxaloacetate, and then the oxaloacetate donates the carbon dioxide to the RUBP with the ribiscotes in order to, of course, go through CAM plants are just going to be temporal isolation. So this would be like your cacti. So at nighttime, they open up their stomata, they bring in all of the carbon dioxide, they fix it in these things called organic acids, and then during the day, they can keep their stomata closed so they can still go through photosynthesis when the light is there, okay? So again, they're going to give you background information. Whew, that gets us through 3. So then let's go back to some of the other questions that were asked. What is an independent variable? Independent variable is going to be a variable that's being modified by the researcher. So they're going to tell you in the experiment that the researcher did this. And because the researcher did this, they modified that thing, right? Your dependent variable is what changed because of that. So because of the fact that they changed this, what did they record? What did they, like, what were they measuring? Okay, what were they finding from it? Okay, and so that would be your dependent variable. And then somebody says, is it always going to be on the x-axis? Nope. Here's the question that's in your CED. This is the FRQ question in the CED. And in this case, your independent variable was actually on your y right here, okay? And so be very, very careful when you're looking at the graph, not to be like, oh, well, whatever's over here on this axis, this must be my independent variable. No, y'all. You got to at least read the question, at least look at it to say, oh, they were determining, you know, how the mortality changed between these different chemicals. I think they were looking at mosquitoes maybe and insecticides. So they were looking at the individuals at different times. And then they were seeing the mortality rate from those. I actually went through this whole FRQ for needing to do this one later, but there's that. It's the one that's in the CED. It's the same one that's in your the Blue Book practice. So, whew. Wait, did I just jump straight into 4? Hey, I just jumped straight into 4. I didn't even give you any warning. Cool. So last chapter, let's get through this. So G protein coupled receptor. Okay, this has to do with it. This is a type of receptor that's found on our plasma membrane. It's going to work with a G protein in order to activate a signal kind of pathway. Okay, and so Here we see the question that was given to us on the, oh gosh, the 2021 exam, I believe. And they gave us this big diagram, and I didn't think that this was going to be clear enough for you to see, so I actually like cropped it and made it even bigger. So here you can see the very first thing, right? So I have an extracellular ligand. There's this ligand, right? And I know that this is going to be this is going to be polar because of the fact that the receptor is sitting on the membrane, right? So the ligand binds to the receptor, okay? And that's going to cause my G protein that is currently inactive to become activated, okay? So the G protein gets the GTP. The GTP then causes it to move over here to my adenylyl cyclase. My adenylyl cyclase becomes activated, which then causes the ATP to be converted to cyclic AMP, which is then going to allow my protein kinase to be activated. And then my protein kinase, as we all know, is going to phosphorylate and allow the phosphorylation cascade. Then that causes there to be chloride released from the cell. And this is what happens in cholera. And this is why individuals that have a cholera poisoning or have cholera, that's why they get dehydrated, because they lose all their water, because as the chlorine comes out, so does the water. So someone said the solutes suck. So as the solute is being pulled out of our cell, we're going to see that the water is also going to run out, okay? So somebody says, is this even English? Yes, this is English, okay? But this is just, you're looking at a diagram and you're pulling all this information out from your diagram. So they're going to ask you a question about it. And later on, I think I even come back to this question to tell you something that they asked about this one. Oh, here's my, see, I told y'all, I'm just like trying to rush to get this PowerPoint done that I have everything in wrong orders. So how secondary messengers amplify the signals, okay? So somebody's like, well, how does phosphorylation cascade, how does all this stuff really make this like whole process, right? So if I have one molecule of epinephrine, right? There's one epinephrine, there's this like hormone that's, fight or flight, all that fun stuff, right? That causes my G protein to be activated. Cool. So now I have 100 molecules of G protein that's activated due to this one epinephrine, okay? So that then activates my adenylyl cyclase, which again is still 100. But those adenylyl cyclases, it's not like they just make one. That was a question on the exam. The one I showed you a second ago was on the exam. So the adenylyl cyclase doesn't just convert one ATP, it converts multiple. And so since I have 100 adenylyl cyclases, now I have 1,000 cyclic AMPs. And those cyclic AMPs, yeah, I know. I told y'all I talk fast. You can't speed me up. Like, I am just fast. Just kind of slow me down. So the adenylyl cyclase is then going to, of course, the cyclic AMP is then going to activate my protein kinase A. Again, we have 1,000 of those. That activates my phosphorylation kinase. It's not really important for you to even pay attention to that. Like, you're just looking at this number right here, right? We go from 10 to the 4, 10 to the 5, 10 to the 6. And look, with one molecule of my epinephrine, I now have 10 to the 8 molecules of glucose 1-phosphate that got released, okay? So that's showing how we're amplifying, okay? The amplification of one signal, you have one single molecule can release multiple secondary messengers that then activates their own individual pathways that lead to a larger response. There's that amplification, okay? So differences between meiosis and mitosis, this is technically unit 5 because meiosis isn't until unit 5, but I went ahead and put it in this thing because I was already building the PowerPoint, so I might as well just do it. So things to focus on is the number of divisions, the daughter cells, and crossing over versus independent assortment on there, okay? So mitosis, we're going to start out with both of them. The parents are diploid. They both go through that one round of replication. I only have, the gene A, I replicate, I make another set of it, okay? And then here's the difference. Mitosis has one division, while meiosis has 2 divisions. So when you look at meiosis, you see like meiosis 1, meiosis 2, prophase 1, prophase 2. Like, this because of the fact that they go through it twice, right? So then when I get done with the whole process, my daughter cells are diploid versus my other daughter cells are haploid. And I don't know what I would do without Miss McClinton answering all your questions in the chat. So let's all love her and give her some thanks in the chat, by the way. So number of daughter cells, we're going to have 2 daughter cells in mitosis, and we have 4 daughter cells in meiosis, right? And so this is due to the fact that I went through one division. So if one cell and I divide once, I get 2, right? But if those 2 cells then divide again, I now have 4, okay? So it just has to do with the fact that we went through 2 rounds of division. That's what led to the 4 cells, okay? In mitosis, they're identical. Nothing happened to separate them and make them different, right? So versus meiosis, they're genetically distinct, okay? We had crossing over, we had independent assortment. All this thing took place in meiosis. Plus, of course, there's only one set of chromosomes, so they're going to be genetically distinct. Every single one of them is different from each other. The difference between haploid and diploid is haploid is going to have a single set of chromosomes versus diploid has 2 sets of chromosomes. The dye makes you think of 2, so diploid. And the reason why they're diploid is because they have a maternal set and a paternal set. They have one from mom and one that came from dad. Okay. And then, as we already said, crossing over does not occur in mitosis or in independent assortment, like doesn't occur in mitosis, but it does occur in prophase, crossing over, and then independent assortment occurs in metaphase 1. So those do take place in meiosis. Okay. Whew. Can I go over action potentials? Action potentials also aren't in the CED, but membrane transport is. So although this probably should have been placed in another section, I think they put it here because of their teacher probably talked about it with cell communication. So you have the sodium. And the sodium is going to be found traditionally outside your cell, and the potassium is usually on the inside of your cell. Okay, and so when a neuron gets a signal that says, "Hey, I lost some stuff in," you're going to see that your sodium channels are going to open. So sodium is going to flow in. So earlier I talked about NANA, right? And so NANA is going to come over to visit. So Na comes into your cell, right? And so that is this depolarization because of the fact that the Na is very positive, right? It's going to cause your membrane potential to become also positive. Okay, and so we have all these sodium channels, like, right, these voltage gated sodium channels, and the sodium just flows into the cell. Cool. So then we close those sodium channels, and the potassium channel opens. Okay, and now the potassium, because we have a high concentration of potassium inside our cell, then it runs out. So the way I tell my students is that your grandma came over, she then pinched your cheek, she asked you about your personal life, she's like, why are you even doing this, right? And she, of course, you run out because you have a car and you're a teenager and you don't really want to be there while grandma's, giving you the third degree. So the K, the potassium, leaves the cell. Okay, so that's where we see this repolarization because of the fact that potassium leaves that cell. Okay, and then at the end we see everything closes, and then what's going to happen is your sodium potassium pump is going to kind of pushes grandma out and brings the kids back in. So it pushes out the sodium and brings back the potassium. Okay, can I please go over independent assortment? That will probably be in unit 5 because that's really where independent assortment and crossing over is. But independent assortment has to do with the homologous chromosomes independently aligned on the metaphase plate. So they can either line up this way or they can line up that way. So each one of them individually can line up on their own. Crossing over has to do with the non-sister chromatids are going to exchange their genetic information. And so if I give me a second, I don't have a pink pen. Okay. So if this was my non-sister chromatids, right? So I've got my maternal and my paternal. Crossing over then allows them to exchange information, so you can have like a new combination of them. Okay. What else? Okay, so I said all that. Cool. So you've seen lots of different signaling transduction pathways, some of them with cascade, some of the G proteins. There's all kinds of ones. Do you actually have to know all the components, all the things? No, you don't really have to know them all. Okay. You need to be able to apply. Okay, so they're going to give you a pathway like what we saw earlier, and they're going to ask you a question about that pathway, and you're going to have to figure it out. And I don't know when part 2 is. I'm trying to figure out if you even want a part 2 of the session, but clearly there's 130 of you in here right now, so I must be doing something right? So here's that one we showed before. I meant to ignore this one, so just ignore the one on the side over here. Like, we don't see that right now, okay? So with cholera, right? There's like they were testing something and they were like, okay, well, what happens if I deactivate the adenylyl cyclase? If I give it some drug, if I give it some way that the adenylyl cyclase cannot work, right? So then what would happen? That's usually what D on your FRQ is, all right? So D is going to be usually where we see like, hey, we broke something. What happens because of that being broken, right? So if adenylyl cyclase cannot get activated, right? We don't have the adenylyl cyclase, okay? Then that means that I don't make cyclic AMP. And if I don't make cyclic AMP, I don't activate protein kinase. And if I don't activate protein kinase, chlorine is not secreted. And this person doesn't lose their water, right? They're not going to see this flushing out and they don't get dehydrated. We don't have all those problems, okay? So now when this question was on the exam, students just said, oh, adenylyl cyclase caused there to be no water secreted. But they never walk through the process. So if they ask you some signaling pathway and they're like, well, what would happen if? Please sure to explain to them every single step and how this thing was broken and how that thing was then broken, or not broken, but like didn't happen. Okay. Over here, we had where I think it was like they inhibited the PMEC. So this is an enzyme, and as you see, the PMEC is going to kind of work to convert the ERK to the PERK. And so they said, what happens if we don't have that? And they asked, like, what would happen to the ratio? Well, the ratio would mean that there's going to be a higher amount of ERK and less PERK, because of the fact that I didn't have the enzyme that was going to facilitate the process of going from ERK to PERK. Okay, and so we would see that the ratio of ERK would increase to a decrease of PERK because like there would just be a larger increase, there would be a larger ratio. Okay, here I don't actually remember what the question asked, and I was running short on time, so I just put the diagram here. But I think what happened was we inhibited the FO85. I really don't remember, but I think that's what happened. This was on 2 years ago, I think, and this caused everybody to want to go after the exam and go have FO. Like, does it make you now want to go have FO? I think so. Anyway, so we inactivated these two. Okay, so what would happen if this wasn't activated? Well, the job of this is to cause your FO4 to get phosphorylated, right? So we phosphorylate our FO4, and if I phosphorylate my FO4, then I don't see that FO4 coming over here to the target genes, right? It inhibits this process. I have no need to do whatever's happening in this pathway, right? So, but if I inhibit this, right, then I no longer phosphorylate that phosphate. Like this is what it normally would have happened, right? We would have phosphorylated, would have stopped, right? But if I stop it, it's similar to what would happen in the low phosphate, where now I'm inhibited and I don't see the FO4. And if I have the FO4 not getting phosphorylated, then now it can bind, which will then allow us to go through and make these target genes, right? So they could ask you a question like that and say, okay, well, this pathway was inhibited. What would happen here? Heredity is in Unit 5. So what's the difference between positive feedback versus negative feedback? Positive feedback has to do with that there's an amplification. I'm building something. I'm making something, right? This is where we see with the oxytocin, right? We build up, pop goes the baby, right? So negative feedback is where we're going to see kind of like a kind of regulation. It brings us back to homeostasis. Most of our things are negative feedback. Most of our stuff is kind of regulating and bringing it back to normal, brings it back to kind of even, right? So we're going to see that our product is going to come back and inhibit the pathway, right? So what we're going to see is that like usually this has to do with like gene expression. And so you're going to see that your product inhibits the enzyme or it'll inhibit the like making of the enzyme. And so that's where we see with our negative feedback. You also see this with like blood and your insulin levels, right? So if your blood level gets too high, right, you're going to release insulin, and then the insulin causes your blood, your cells to take up the blood sugar, which then decreases it. Like, so that would be negative feedback also. But usually when we talk about in bio, we're usually talking mostly about like gene expression. Explain cyclin and CDK as part of checkpoints. So cyclin is a protein that's synthesized during the cell cycle. There are multiple different types of cyclin. I mean, you can kind of see like their concentrations, and they do vary when they happen. And then cyclin-dependent kinase is a kinase, so it's of course responsible for phosphorylating. And then the cyclin-dependent, meaning that it depends on cyclin. Okay. Now you don't need to know all these specific cyclins. They're going to give you some type of diagram. They'll probably give you a prompt. They're going to give you something that makes you say, oh, this is what's happening. Okay. So here I've got this no cyclin, so my CDK is inactive. And so since the CDK is inactive, it's no longer going to phosphorylate these targets. And so since it doesn't phosphorylate these targets, I don't go through the S phase. And of course, we know the S phase's job is for us to, of course, synthesize the DNA. Okay, so I don't go through that process. Okay, versus if I do have my cyclin and my G1 and my S cyclin, right, those come through, they bind to the CDK, and that then phosphorylates these targets. So since these targets have been phosphorylated, they then cause you to go through the DNA replication. It kind of allows for that checkpoint to be overcome to be able to do something, okay? So the cyclin is required for us to get through this. And so like there's usually like a buildup of the cyclin until it finally gets to an appropriate concentration. And at that concentration is usually when you're able to bypass that checkpoint that's needed, right? So do you need to know about receptor tyrosine kinase? I don't know. One of my textbooks has it. One of my textbooks doesn't. So I don't really know if you need to know. But again, you're going to be given some type of prompt, I assume. They're going to say, hey, there's this receptor. And you might be able to see the picture. You might not be able to, but it doesn't really matter. So here with our tyrosine receptor kinase, we've got all these tyrosines on it. And their job is, of course, kinase. They're going to phosphorylate. So when I bind, so this is EGF, I think it's probably some type of growth factor. And so when you bind to it, then it phosphorylates. And so that can then go through multiple pathways from there. So how do you answer a feedback question? I don't know, follow the pathway. So this question said, explain one feedback mechanism by which a cell could prevent production of too much IAA without inhibiting I3PA production, right? So here they gave us this pathway. They showed us that you have the tryptophan. We use the enzyme TRPT that then makes I3PA. The I3PA uses the enzyme YUC to make IAA, right? So I want to regulate this, right? I want to limit how much of this is made, but I don't want to mess with this, okay? which means that this whole pathway right here is not going to get touched. Okay, I don't want to mess with this one because I need I3PA to stay the same. So instead, I can modify this. Instead of going through the gene to mRNA, instead of making this enzyme, then I won't be able to go through IAA. This stays the same, and IAA will be regulated because of the fact that there's not as much of the enzyme to bind up. Yes, I will post a video later. So we also could just inhibit the enzyme. So if if the IAA binds to the yuck enzyme, then that could also slow down or decrease the production because it can inhibit it, right? Yes, limit the yuck. You can't delete the mRNA. You just don't make the mRNA. You inhibit the transcription, so you don't make the mRNA, which means you don't make the protein. Yeah, it's okay. So P53. So P53 is a tumor suppressor gene. So you have two different ones. You have tumor suppressors, and then you have your oncogenes. Tumor suppressor, their job is to inhibit excessive growth, versus your oncogenes are normally it's a proto-oncogene that's like our normal cell division processes, and they get mutated and then they become overactive. So like the RAS protein would be an example. Okay, and so in this example, looking at our tumor suppressor, looking at the P53, there was some type of damage because of the UV light, and so because of that, these protein kinases, they activate the P53. P53 then makes this certain protein that says, "Hold up, wait a minute, put a little bio in it," and it stops the cell cycle. Okay, and so since there is a problem with my DNA, I don't want the cell to replicate. I don't want to make more copies of the cell because there's an error, right? So I'm so sorry, the diagrams make no sense. And so we're going to stop the cell cycle until I can fix this DNA damage. So since I'm waiting to fix the damage, right, it's holding up. But if there's a mutation and I don't have P53, then I don't make the thing to stop the cell cycle. And I'm going to see that I'm going to keep on replicating. I'm going to make copies of the cell that has damage in DNA, which is a major problem, right? And then there's no one about the proto-oncogenes to oncogenes, but the overall thing is that I've got like this normal cell division thing. It's like, oh, cell should divide, cell should divide. And so if there is an error in that, and I'm making too much of that cell should divide, then it causes the cell to grow too much, okay? So explain phosphorylation cascade, has to know when to stop and start. All these little intermediates get phosphorylated, and so it knows to stop because it reaches the end. Like, I don't really know what to tell you. Every single pathway is different. Every cell, even if it's the same exact signaling molecule, the different cells are going to have different responses to the same signaling molecule. So there's no way to know them all. And again, they're going to have to give you some information. There's really no keywords to memorize in this. If you're wanting to know keywords for FRQs, check out the CED. They tell you exactly what all of their like task verbs mean. And I'm gonna try to get my FRQ Friday this week to be about like FRQ tips and tricks. So I should be posting that Friday if I get my life together. But clearly it took me 5 hours to make this, so who knows? How do the checkpoints in cell cycle work? Where are they, and how do the cells know that they can move on? How do they mess ups happen? So like there's 3 different checkpoints that we talk about. The G1 is like the go ahead signal. Like if you get a signal at the G1, you go ahead and actually go through cell division. This tells you, hey, it's time for the cell to actually grow and divide. G2 is like at the end of the S phase. We've gone through S phase, we've gone through S2. Did it make sure that the cell grew big enough? Did I have where I've got my my DNA has been replicated? There's no errors in my DNA. Like, did it grow like it needed to grow, right? And then the M signal makes sure that I have every single kinetochore microtubule attached to every single chromosome, so that I have equal division in anaphase. Okay. Are ligands proteins? No, not always. So ligand is just your signaling molecule. So if it's a protein, it's going to have an extracellular receptor. If it's a hormone, it's going to have an intracellular receptor. And then can you explain autocrine, paracrine, endocrine? Auto makes us think of me, so self. So autocrine is going to bind to the same cell. Paracrine means it's going to bind to a cell nearby or next to, okay? And then endocrine usually gets into the bloodstream. It travels all over the bloodstream until it finds the appropriate cell. Which ones are the appropriate cells? The ones with the receptor. So if it has a receptor for that signaling molecule, then it's going to be able to bind to then go through it. Lac operon is going to be a inducible operon. Okay, so here you can kind of see it. Ignore the direct signaling because that's just talking about like, one to the other, like binding to each other, so like cell communication that we talked about at the beginning. So paracrine signaling, it releases out and it binds to the receptor on a nearby cell. Autocrine, we see that it's going to bind to like its own cell, so it releases out and it binds to itself. And then this is a type of kind of like paracrine. This is a neuron. You're not required to know about neurons, but like this is, they could talk about a neuron and talk about like the synapse, and they might mention it, but they have to give you all the information for you to be able to say, oh, this signaling molecule, which is called a neurotransmitter, binds to the receptor that's on this target cell. And then here's endocrine. It's going to be released out into the bloodstream, and then it binds to a target cell. So that is the end of 4. So then how do I know when to use SEM and error bars? Well, traditionally the SEM is your error bar. So when you're looking at your graph and you've got these error bars, that's usually your standard error of the mean. And the way that they write it out is SE, and then there's a little X bar. So like SEX, yeah. Anyway, so the error bars demonstrate the variation of the data. And so that's just your definition you have of your standard error of the mean, okay? So we go back to this question. You say, okay, well, Ms. Jones, I graphed this. How do I know how to graph my error bars? So they tell you, you do not have to calculate it. It's right here. Oh, thank you so much, Alec. So we got this plus or minus. So when I graph this, I'm going to say, okay, well, 12.8 is my mean. So here's the top of my bar, because we said earlier this was a bar graph, right? And then I'm going to go up 2.2 and make the top of my error bar. And I'm going to go down 2.2 and make the bottom of my error bar. And I will do that for each of these values. I'll go 16.5 plus 2 minus 2, 22.1 plus 0.7 minus 0.7. And I'm going to do each of those whenever I graph it, okay? So here you can kind of see what that graph would look like. So here would be those standard errors of the means. You see they went above and below the values that we just mentioned, okay? And this is an example of that graph we talked about earlier. When you're graphing them, make sure that you don't cover up the lower bound. And as I said, we don't see colors, right? And so this person could have used a blue and a black ink, but we would have no idea, right? So you're running out of time and you start blanking, what's the best way to get the most points? Go with your gut. Traditionally, your first answer is going to be the right answer. Like if you read a question like, oh, this is the answer, boom, that's usually going to be the answer. Like you got to trust yourself, y'all. Like you only got a minute and a half per question. Just trust yourself. You are so smart. You are Penguins, you're dressed for success. You know all this information. Don't let the timer get you stressed. Don't let the fact that this is some big high-stakes exam get you stressed. Y'all, it's a test that you're taking on one day. Do not let it stress you out. Like just show them all the bio you've learned this year. Show them all the hard work you've put in. Like just show off. Okay. So first thing is your first answer is probably gonna be your answer, right? Strategy. So as I'm thinking about it, like logically back in the day, we used to be able to, oh, there's 1 minute left. Let me just quickly bubble some stuff in. You're not going to have that option to be able to go through and pick out, hey, I haven't answered these questions. So my advice is that if you're planning on skipping something, just go ahead and mark the answer that you think is the right answer or your first guess, right? And then mark it to come back to, mark it for review so you can come back to it, okay? If you have time at the end, you go back. If not, then at least you have an answer marked, okay? And then use your resources. A lot of these questions are going to have diagrams. They have prompts. Take a deep breath. You know this information and just figure it out, right? If they're asking how does one thing affect another, look at the diagram. How do I see a correlation between these two things? Do not breathe. I mean, like, do breathe. Don't freak out. So what's the advice for finishing your FRQ if you're struggling on one of the questions? Easy pointers. So easy points are like things that you definitely need to answer because of the fact that like you don't want to lose points on this kind of thing, right? So first, graphs, right? The graph is usually worth 3 points. The whole FRQ section is only like 30 something points, y'all. Like it's 18 plus 16, what's that, 34? Like it's only 34 points. Like so there's a total of 3 points just in your graph, and 1 point is usually analysis of that graph. So like literally make sure you do your graphs, okay? If they ask you to predict something, y'all, it doesn't take that hard to predict something. If you're wrong, who cares? You at least tried. Do not leave a prediction question blank, okay? Question 5, number C, is always talking about, like, it's a diagram. You're going to have to annotate some diagram. You have to do something, right? 1 year, it was adding an invasive species to a food web. Like, seriously? Oh, come on. Or it was circle something. Like, if all they're asking you to do is circle something, put an X on something, like, y'all, that takes 5 seconds. Don't skip that. At least answer it, okay? And then experimental design. You don't have to know anything about biology to know what's an independent variable, what's a dependent variable, what's a control, right? Why would you use a control? You at least can try to guess on that information. You don't have to know. It's okay if you don't. You at least got to try. And then, as I said earlier, answer in the order that you know the answers, right? So if you're really confident about C, answer C. If you're really confident about B, answer B, okay? It doesn't matter if you're in the exact same order, okay? You can on your paper go C, A, D, be. Like, you can go in any order you want to go. Okay. Answer them in the order you know them, label them so it's easier for your readers to find your answers. But you can answer them in any order. So answer what you know first and then come back to what you don't know. Don't sit there staring at a question that, like, oh, well, I don't know. I don't know. Let me stare at the sun. Like, no, answer what you know and move on. Okay. And then what is that? Okay, so if you're graphing, so they were asking about the annotations. So your FRQs are going to be given to you, or they're going to be accessed on a computer. So you're either going to have an iPad or a computer on your Blue Book app, and on there you're going to see the question. And then in your book, you're just going to have a bunch of lines. I think I showed it earlier when I said like how to respond. But if you want to see the example book, it's on AP Central. If you go to AP Central, like it's on the main page, it says like, sample response book. Okay. And then they said, is there going to be different versions or all the students the same version? There are, I think, God, like 4 or more different versions of the exam. They're not like making more versions because of it being online that I know of. I could be wrong. But they are going to have different versions of the exam. And so they're not changing the exam for any other reason but the security. By having it be a hybrid exam, by them regulating when you access the tests, they can ensure that there's not going to be a leak of the questions. They can ensure that there's no issues in terms of some students having access too earlier or after another. So that was the session. I'm so sorry that I went way over an hour. But as I said, I wanted to make sure I got you as much as I could. This was all that I could get together in my 5 hours. I did my best. I'm going to try to do another session. I asked earlier and you all were like massively saying, please do a second session. So on my Instagram today, like right now in my story, there's a question. It's asking, should I keep my question boxes up? Should I use the time I usually do on my question boxes to go ahead and put together the PowerPoint? And then maybe I can get this put together like one evening. The sooner that I can put the PowerPoint together, the sooner I can do it, because I do not want to last minute be doing a session. Like, there is no part of me. There's a reason why I'm doing this a week before the exam. I want you to have everything you have to be slowly able to review and not be cramming. I do not want you to study on Sunday the 4th. I want you to spend it with your family. I want you to do something fun. I want you to be a teenager. Okay. This is one test on one day. Do not freak out. But as soon as I know when I'm doing that other session, or if I'm doing the other session, I'll post it on my Instagram. So if you're not already following AP Bio Penguins, be sure to follow it. If you want to, like, as soon as I figure out the date, I'll probably post on my YouTube page. I'll have like the link ready to go. So you can easily go and say, oh, look, Miss Jones like went ahead and posted like when this live's going to be. And the goal is that I'm going to do the live early enough that even if you can't catch it live, you're going to be able to access it later. This PowerPoint will be posted on my website. You'll be able to get to it. Like, I'm going to do everything I can to get this to you as soon as possible, but it took me 5 hours just to put this together, and I fear that the later units might actually have more questions in them. I don't know. And any questions that were in there posted in the wrong unit, I'm probably not going to go back and redo those just because this is the session. This is over. In case you don't know and you're looking for more resources, because I'm killing time because I'm trying to make sure that we, get to the end of the live. This is the website, apbiopenguins.weebly.com. If I post on my website, I don't know if you're really going to see it, but I'm happy to post on my website. Like, this is where I've been posting everything else. If you go here, the review calendar, you can see like these are all the other previous sessions. Clearly, I didn't get Unit 8 made, but it was made. It's just not there yet. But like, you could easily come here and I'll have the next one posted, ready to go. But on the website, in case you're like, I need resources, this is what was helpful last year. There's unit review videos. Like Michelle, I really need more help on Unit 5. Like, go watch the unit review video. There's TikTok videos on every single topic. There's 120 quizzes, games. If for some reason, like, my codes down here don't work, quizzes like reached out and they're like, hey, can we put a Google Doc together with all your stuff? I'm like, sure. So if you go here, you actually have every single one of my games where you just like click this button and you can play it as a kid. Like, they will not ask you for code. You just click it and go. It's great. But that's tons of resources. If you're like, I need to figure out last minute, like, hey, how can I be successful? This is the data from last year's kids. Where is it? There's a document. Oh, right here. If you download this file, you can say, hey, this is what the 5s did. This is what the 4s did. You can see exactly what you need to do to be successful in the exam, or your version of success. For me, my version of success is that you did your best. Like, I don't care if you make a 1, 2, 3, 4, or 5. I'm very proud of you no matter what score you get. And I just want you to know that this is one day, one test, and you are a penguin and you are dressed for success. So with that, I'm going to tell you that I hope you have a good